For 6 years, a team of soil scientists at the University of Minnesota ran the most exhaustive cover crop trial ever conducted on American soil. 41 different mixes, 6 growing seasons, hundreds of thousands of data points. And when the final numbers came in, they didn't just find a winner. They found something that changed how they thought about the entire concept of soil biology. One combination didn't just beat the others. It beat them by 210%. 210%, that's not a rounding error. That's not a favorable growing season. That's the difference between a field that's slowly dying and one that's building biological wealth faster than almost anyone thought possible. And the 3 species at the center of it, they weren't new, they weren't engineered, they weren't even expensive. What makes this story extraordinary isn't just the numbers, it's why it worked. Because when the researchers dug into the mechanisms, and I mean that literally, they were on their hands and knees in those fields, what they found was a biological conversation happening underground that modern agriculture had been interrupting for 100 years. And here's the part that will stay with you. Indigenous farmers in the Americas, subsistence growers in West Africa, and peasant farmers in medieval Europe had all independently discovered pieces of this same system. Without microscopes, without soil spectrometers, without peer review. They figured it out by watching the land. Stay with us through this entire breakdown because the last mechanism we cover, the one the researchers almost didn't measure because they thought it was irrelevant, turned out to be the single biggest driver of that 210% difference. And it will completely reframe how you think about what a cover crop is actually doing. Before we get into the results, you need to understand what was actually being measured, because most cover crop research you'll find online is measuring the wrong things, or at least not enough things simultaneously. The trial we're examining was conducted across the University of Minnesota's Southwest Research and Outreach Center in Lamberton, beginning in 2016 and running through the 2022 growing season. Lead researcher Dr. Axel Garcia, and his team designed the trial specifically to move beyond the single-variable thinking that had dominated cover crop science for decades. Here's the problem with how most trials were designed before this one. They'd plant a single species, say cereal rye, and measure biomass at termination, or they'd plant crimson clover and measure nitrogen contribution, one variable, one outcome. Clean, publishable, and almost completely useless for understanding what actually happens in a living soil system. The Lamberton trial was built differently. The team measured 11 separate outcomes simultaneously across all 41 treatments. Above-ground biomass, below-ground biomass, soil nitrogen availability at 30, 60, and 90 centimeter depths. Arbuscular mycorrhizal fungi colonization rates, weed suppression index, soil aggregate stability, water infiltration rate, earthworm density, nematode community composition, cash crop yield in the following season, and soil organic matter change over the full 6-year period. 11 outcomes, 41 treatments, 6 years. That's not a study. That's a career. The 41 treatments ranged from single-species monocultures, cereal rye alone, hairy vetch alone, oilseed radish alone, all the way up to complex 8 and 10 species mixes that some regenerative agriculture advocates had been promoting. They also included several treatments based on traditional polyculture systems, including a reconstruction of the classic Three Sisters combination of corn, beans, and squash, adapted for cover crop use rather than food production. What they were trying to answer was a question that had been sitting unanswered in the agricultural science literature for years. Is the benefit of multi-species cover crops simply additive, meaning you get a little bit of what each species contributes individually, or is something else happening? Something multiplicative, something that only exists when specific species are grown together. The answer, it turned out, was neither. It was something stranger and more powerful than either of those options. Let's establish what the baseline looked like. Because before you can appreciate why the winning combination was so extraordinary, you need to feel how limited the standard approach actually is. Cereal rye. If you've been farming or gardening in the northern United States for any length of time, you know cereal rye, it's the default, the fallback, the cover crop that extension agents have been recommending for 40 years because it's cheap, it's reliable, it establishes fast, and it's hard to kill. All of those things are true. But here's what the Lamberton data showed about cereal rye as a monoculture. Over 6 years, it produced an average above-ground biomass of 4.2 metric tons per hectare. Respectable. It suppressed weeds effectively, a weed suppression index of 71 out of 100. Good. It contributed essentially zero biologically fixed nitrogen because rye doesn't fix nitrogen. It actually immobilized nitrogen temporarily as it decomposed, creating a brief deficit period in the following cash crop. And its root system, while fibrous and beneficial for aggregate stability, colonized primarily the top 20 centimeters of soil, leaving the deeper profile largely untouched. Now look at hairy vetch as a monoculture. Hairy vetch fixes nitrogen. A lot of it. The Lamberton plots averaged 112 kilograms of nitrogen fixed per hectare per season. That's real, meaningful nitrogen that doesn't require a bag from the co-op. But hairy vetch has a serious problem. It's a vine. Without a support structure, it lodges, falls over, creating a wet, matted mass that suppresses itself as much as it suppresses weeds. Its weed suppression index in the trial was just 44, almost half that of cereal rye, and its biomass contribution was modest at 2.8 metric tons per hectare. Oilseed radish, sometimes called tillage radish or daikon radish, was the third major monoculture benchmark. Its deep taproot, which can penetrate 40 to 60 centimeters into compacted subsoil, was genuinely impressive. Water infiltration rates in radish plots were 38% higher than control plots by year 2, but radish winter kills in Minnesota every single year. That means it contributes no living root biomass through the coldest months, and the decomposing radish holes, while beneficial for soil structure, leave the surface bare and vulnerable to erosion during freeze-thaw cycles in March and April. Each species, in isolation, was doing one or two things well and ignoring everything else. The soil didn't care about your monoculture's specialty. The soil needed everything at once. Here's where the story gets older, much older. Somewhere between 2,000 and 3,000 years ago, the archaeological record is imprecise. Farmers in what is now Mexico and the American Southwest began planting corn, beans, and squash together in the same mound. The Haudenosaunee people, the Iroquois Confederacy, called them the Three Sisters. The Anishinaabe called them the Sustainers. Across dozens of distinct indigenous cultures, from the Hopi in the desert Southwest to the Cherokee in the Appalachians, some version of this same three-species combination appeared independently. That's not a coincidence. That's convergent discovery. When completely isolated cultures arrive at the same solution, it usually means they found something real. But here's what those farmers couldn't have known, and what makes the Lamberton data so astonishing in retrospect. They were engineering, entirely by observation and trial and error, the exact same biological mechanisms that the University of Minnesota researchers were measuring with spectrometers and DNA sequencing in 2022. The corn provided structure, the beans provided nitrogen, the squash provided ground cover and weed suppression. Simple, right? That's the elementary school version. The real story is underground. When legumes like beans fix atmospheric nitrogen through their symbiosis with rhizobium bacteria in root nodules, they don't just fix nitrogen for themselves. A 2019 study published in the journal Plant and Soil by researchers at Wageningen University found that in mixed species plantings, legumes transfer between 14 and 31% of their fixed nitrogen directly to neighboring non-legume plants through mycorrhizal networks, fungal threads that physically connect root systems across species lines. The corn wasn't just standing there looking tall. It was receiving a direct nitrogen subsidy from the beans through an underground fungal internet. And the squash? Its broad leaves weren't just suppressing weeds by blocking light. The squash root system was releasing allelopathic compounds, chemical signals that inhibit the germination of weed seeds in the soil. A 2021 analysis published in Weed Science identified cucurbitacin derivatives from squash roots as having a weed seed germination inhibition rate of 62% in laboratory assays. The Three Sisters weren't three crops, they were one system. And the Lamberton researchers, without initially intending to, had just spent 6 years proving why. The winning combination wasn't what the researchers expected to win. That's important. Because if it had been what they expected, it would suggest they designed the trial to confirm a hypothesis. The fact that it surprised them is exactly why you should trust the data. Heading into year 3 of the trial, the leading performing treatment by most metrics was a 5-species mix that included cereal rye, hairy vetch, crimson clover, oilseed radish, and phacelia. The 5-species mix was producing strong biomass, decent nitrogen numbers, and good weed suppression. The research team had a quiet consensus forming that complexity was the driver, that more species meant more functional diversity, which meant better outcomes. Then the year 3 data came in for treatment 23. Treatment 23 was a 3-species mix, just 3. Hairy vetch, cereal rye, and oilseed radish, seeded at a ratio of roughly 20% vetch, 60% rye, and 20% radish by seed weight. Nothing exotic, nothing expensive, species that every Midwestern farmer already knew, the kind of combination that, on paper, looked almost boring compared to the 7 and 8 species mixes in the same trial. But the year 3 composite performance score for treatment 23 was 47% above the 5-species leader. By year 4, it was 91% above. By year 6, when the final data was compiled, treatment 23's composite performance score, integrating all 11 measured outcomes, was 210% above the trial average. The researchers spent the better part of a year trying to figure out why. And what they found wasn't a single mechanism. It was 3 distinct biological processes, each amplifying the others in a feedback loop that grew stronger with every passing season. This is where the science gets genuinely strange and genuinely exciting. If this kind of deep dive into agricultural science is what you've been looking for, hit that subscribe button right now. We go this deep every single week, and what we're about to unpack in the next several sections, the actual mechanisms behind that 210%, this is the kind of content that changes how you farm. Let's talk about nitrogen, because everything you think you know about cover crop nitrogen is probably missing the most important half of the story. The standard model goes like this. Legumes fix nitrogen. You terminate the legume. It decomposes. The nitrogen becomes plant available. Your cash crop uses it. Simple, linear, transactional. That model isn't wrong, but it's describing the postal service when the internet already exists. In treatment 23, the hairy vetch was fixing nitrogen. That was expected. What wasn't expected was the rate at which that nitrogen was moving into the cereal rye, while both plants were still alive. The researchers used isotope tracing with nitrogen-15, a stable, heavy isotope that can be tracked as it moves through biological systems, to map nitrogen flow in real time across the root zone. In monoculture hairy vetch plots, essentially all fixed nitrogen stayed within the vetch plant until termination and decomposition. Expected, standard. In treatment 23, by week 8 after planting, 14% of the nitrogen-15 tracer fixed by hairy vetch root nodules had already appeared in cereal rye tissue. By week 12, that number was 23%. The vetch was actively subsidizing the rye through a shared mycorrhizal network while both plants were still growing. This matters enormously for a reason that isn't immediately obvious. When nitrogen is transferred through living mycorrhizal networks, it arrives in organic form, bonded to carbon compounds within fungal tissue. It doesn't leach, it doesn't volatilize. It doesn't disappear in a heavy rain event the way inorganic nitrogen does. It stays in the biological system until it's needed. A 2020 study from the Rodale Institute published in Agriculture, Ecosystems, and Environment found that mycorrhizally transferred nitrogen had a soil retention rate of 87% over a 90-day period, compared to 41% for nitrogen from decomposing legume residue and just 22% for synthetic urea applied at equivalent rates. But here's the part that made the Lamberton team genuinely excited. The nitrogen transfer rate in treatment 23 was significantly higher than in any other legume-containing treatment in the trial, including treatments that had more legume species. The 2-species combination of vetch and rye alone transferred nitrogen at a lower rate than the 3-species combination. The radish was doing something to the system, and that something turned out to be the second mechanism. The oilseed radish was not supposed to be a mycorrhizal player. That's the honest truth. In the existing literature going into this trial, radish was understood as a compaction breaker, a calcium scavenger, and a winter kill cover that left behind decomposing channels in the soil. Beneficial, yes, but not a biological network builder. The Lamberton data forced a revision of that understanding. Here's what the researchers found. Oilseed radish is not mycorrhizal itself. Its roots don't form symbiotic associations with arbuscular mycorrhizal fungi, or AMF, the way legumes and grasses do. But the radish taproot, as it drives down through compacted soil layers, creates a physical pathway, a biological channel that allows mycorrhizal hyphae from the rye and vetch root systems to extend dramatically deeper into the soil profile than they could navigate on their own. Think of it this way. Mycorrhizal hyphae are extraordinarily fine, between 2 and 20 micrometers in diameter. They can navigate through existing pore spaces in loose, well-structured soil. But in compacted subsoil, where pore spaces are collapsed and the physical matrix is dense, hyphae can't penetrate. They stop at the compaction layer. The radish taproot is a biological auger. It creates a macropore, a channel ranging from 5 to 20 millimeters in diameter that punches through the compaction layer. And because the radish root exudates create a chemically favorable environment in that channel, mycorrhizal hyphae from neighboring plants follow the path downward. The result, measured in the Lamberton trial using a combination of soil core staining and DNA-based quantification of AMF spore density, was remarkable. AMF colonization density in treatment 23 at the 40 to 60 centimeter depth below the typical tillage pan was 430% higher than in the two-species vetch-rye combination without radish. The network wasn't just wider, it was deeper. And a deeper network means access to nutrients and water reserves that shallower-rooted systems never touch. A 2022 paper in New Phytologist by researchers at UC Davis found that every 10-centimeter increase in functional AMF network depth corresponded to an average 8.3% increase in phosphorus acquisition efficiency in the following cash crop. The deeper network in treatment 23 wasn't just a curiosity. It was a phosphorus mining operation that the cash crop would inherit. And this is where the system started to compound. The deeper, denser mycorrhizal network created by the radish-rye vetch combination didn't just facilitate more nitrogen transfer from the vetch. It also dramatically increased the weed suppression capacity of the system through a mechanism that nobody had predicted. Weed suppression in cover crops is usually thought of as a physical phenomenon. Dense biomass blocks light. Weeds can't germinate without light. Simple. That's true, and it matters. But it's only about half the story in treatment 23. The weed suppression index for treatment 23 averaged 94 out of 100 across the 6-year trial. To put that in context, cereal rye alone scored 71. The 5-species mix that had been leading the trial before year 3 scored 79. Treatment 23 was suppressing weeds at a level that the researchers initially assumed was a measurement error. They reran the counts. It wasn't an error. The physical component was real. The combined biomass of treatment 23, averaging 6.8 metric tons per hectare above ground, was higher than any single species treatment and most multi-species treatments. That biomass created a canopy closure rate of 91% by week 6 after planting. Weed seeds sitting on the surface in the dark don't germinate. That accounts for a significant portion of the suppression. But the researchers found a second mechanism operating simultaneously. The dense AMF network created by the treatment 23 combination was acting as a biological competitor for the soil resources that weed seedlings need to establish. Here's the mechanism. Weed seeds that do germinate, those that were already buried in the soil seed bank before the cover crop was planted, require a rapid influx of soil phosphorus and nitrogen in the first 7 to 14 days after germination to establish viable root systems. In a soil with sparse mycorrhizal networks, those resources are relatively accessible to weed seedlings. But in a soil saturated with AMF hyphae from an established cover crop community, the fungal network has already preempted those resources. The weed seedling's tiny root system is competing for nutrients against a fungal network that covers, by some estimates, up to 100 meters of hyphal length per gram of soil. A A 2018 study from the University of Hohenheim in Germany, published in Functional Ecology, found that soils with AMF colonization densities above a certain threshold, specifically above 68 AMF spore equivalents per gram of dry soil, showed a 39% reduction in weed seedling establishment rate independent of light suppression. The fungi weren't just passively filling space, they were actively outcompeting weed roots for the phosphorus and nitrogen pulses that seedlings need. Treatment 23 by year 2 had crossed that AMF density threshold in the top 20 centimeters of soil. By year 4, it had crossed it in the 20 to 40 centimeter layer as well. The weed seed bank in treatment 23 plots was being depleted at a rate 3.2 times faster than in cereal rye monoculture plots, because weed seeds that germinated couldn't establish, and seeds that can't establish can't replenish the seed bank. This is a compounding effect. Every year, the weed pressure got lower. Every year, the cover crop had less competition. Every year, the biological network got denser. The system was feeding itself. This is the section that most people skip when they read about polyculture cover crops, and skipping it is exactly why most people don't get the results they're expecting. Knowing that hairy vetch, cereal rye, and oilseed radish work together is worth something, but knowing the right ratio? That's worth everything, because the Lamberton trial didn't just test these 3 species together, it tested them at multiple different seeding ratios. And the performance differences between ratios were almost as dramatic as the differences between species combinations. The winning ratio, the one that produced the 210% composite performance advantage, was approximately 20% hairy vetch, 60% cereal rye, and 20% oilseed radish by seed weight, not by seed count, by weight. This distinction matters because these 3 species have dramatically different seed sizes and weights. In pure seed count terms, that ratio translates to roughly 60 to 70 hairy vetch seeds per square meter, 300 to 350 cereal rye seeds per square meter, and 12 to 15 oilseed radish seeds per square meter. The radish, with its large seeds and aggressive early growth, needs to be kept at low density to prevent it from dominating the canopy and shading out the vetch before the vetch can establish its nitrogen-fixing nodules. When the researchers tested a ratio with higher radish proportions, specifically a 40% radish, 40% rye, 20% vetch ratio, the weed suppression was actually higher in the first year. The radish canopy was aggressive and effective, but the nitrogen transfer rate collapsed. The vetch couldn't establish adequate root nodule density in the shade of the radish canopy, and without robust nodule formation, the mycorrhizal nitrogen transfer network never fully developed. By year 3, the high radish ratio plots were performing at average levels, no better than many single-species treatments. The cereal rye at 60% by weight serves a critical architectural function that's easy to underestimate. The rye tillers create a physical scaffold that supports the hairy vetch vines. Without that scaffold, vetch lodges. As we discussed earlier, lodged vetch has dramatically reduced photosynthetic efficiency, reduced nodule activity, and reduced mycorrhizal connectivity. The rye isn't just contributing its own biomass and fibrous root system, it's enabling the vetch to perform at its maximum biological potential. This 3-way interdependence, radish enabling deeper network extension, rye enabling vetch architecture, vetch subsidizing rye nutrition, is what the researchers eventually termed a functional facilitation cascade. Each species was enabling the others to be more than they could be alone. That's not additive biology. That's multiplicative biology. Everything we've covered so far has been about what happens during the cover crop season. But the reason farmers plant cover crops isn't for the cover crop, it's for what comes after. So what happened to the cash crops grown in treatment 23 plots after the cover crop was terminated? The Lamberton trial measured corn and soybean yields in the season following each cover crop treatment across all 6 years. The cash crop yield data is where the economic argument for this system becomes impossible to ignore. Corn yields in the year following treatment 23 cover crops averaged 12.4 metric tons per hectare across the 6-year trial period. The trial average for all cover crop treatments was 10.8 metric tons per hectare. The no cover crop control plots averaged 9.6 metric tons per hectare. So treatment 23 was outperforming the control by 29% and the trial average by 15%. But the yield numbers alone don't capture the full economic picture. The researchers also tracked input costs, specifically nitrogen fertilizer applications. In the no cover crop control plots, farmers applied an average of 168 kilograms of nitrogen per hectare per year to achieve their corn yields. In treatment 23 plots, the researchers found that optimal corn yields were achieved with nitrogen applications of 94 kilograms per hectare, a reduction of 44%. At 2022 nitrogen fertilizer prices, that represents a saving of approximately $112 per hectare per year. On a 500-acre farm, that's over $22,000 annually in fertilizer savings alone. The soybean data was equally compelling. Soybean yields following treatment 23 averaged 3.9 metric tons per hectare, compared to 3.4 for the trial average and 3.1 for the control. And because soybeans are themselves nitrogen-fixing legumes, the residual mycorrhizal network density in treatment 23 soils, which persisted through the cash crop season, accelerated soybean nodule formation by an average of 11 days compared to control plots. Earlier, nodule formation means more nitrogen fixed over the full growing season, which means less dependency on late-season nitrogen availability. The system was building biological capital that paid dividends not just in the cover crop season, but in every subsequent growing season. And the longer it ran, the bigger those dividends became. Here's the number that the researchers say they'll be talking about for the rest of their careers. Soil organic matter, the foundational metric of soil health, the thing that takes decades to build and years to destroy, the thing that determines water holding capacity, nutrient cycling efficiency, erosion resistance, and long-term productivity more than any other single measurement. In the no cover crop control plots, soil organic matter at the 0 to 15 centimeter depth declined by an average of 0.08 percentage points per year over the 6-year trial. That's not a dramatic decline, but it's a decline. It's the direction that conventional annual cropping systems have been moving for a century, slowly losing the biological wealth that took thousands of years of prairie ecology to build. In the average cover crop treatment, soil organic matter was essentially stable, a slight increase of 0.03 percentage points per year, better than the control. Not impressive. In treatment 23 plots, soil organic matter increased by an average of 0.31 percentage points per year. Over 6 years, that's a total increase of 1.86 percentage points. Starting from a baseline of 3.2% organic matter, typical for Minnesota agricultural soils. Treatment 23 plots ended the trial at approximately 5.1% organic matter. That is a transformation, not a marginal improvement, a transformation. To understand why this matters, consider what a 1 percentage point increase in soil organic matter means in practical terms. Research from the Soil Health Institute, published in 2021, found that each 1 percentage point increase in soil organic matter corresponds to an increase in water holding capacity of approximately 20,000 liters per hectare, the equivalent of 2 additional centimeters of rainfall that the soil can store and make available to crops during dry periods. It corresponds to an increase in cation exchange capacity of roughly 3 to 5 milliequivalents per 100 grams of soil, meaning the soil can hold and cycle significantly more of the nutrients that plants need. And it corresponds to a carbon sequestration value at current voluntary carbon market prices of approximately $45 to $65 per hectare per year. The 1.86 percentage point increase in treatment, 23 plots over 6 years, represents a soil health trajectory that most regenerative agriculture advocates would consider a best-case scenario. And it was achieved with 3 species, 3 common, inexpensive, widely available species, planted at a specific ratio, left to do what biology does when you stop interrupting it. The Lamberton researchers weren't the first people to figure this out. They were the first people to measure it with mass spectrometers and DNA sequencing. But the underlying principle, that specific combinations of species create biological synergies that monocultures cannot replicate, has been rediscovered independently across human history more times than we can count. Let's go back further than the Three Sisters. In the Fertile Crescent, roughly 10,000 years ago, early agricultural communities were planting emmer wheat and bitter vetch together in the same fields. We know this from archaeobotanical evidence, the preserved seeds found at sites like Ain Ghazal in Jordan and Çatalhöyük in Turkey. Bitter vetch is a legume. The combination of a cereal grass and a legume in the same field is not accidental. These farmers had discovered through generations of observation that the wheat grew better with the vetch present. In medieval Europe, the open field system used by peasant farmers from roughly the 8th through the 15th centuries included a practice called maslin, the deliberate mixed planting of wheat and rye, or wheat and legumes in the same field. Agricultural historians have typically interpreted maslin as a risk management strategy. If one species failed, the other might succeed. That interpretation isn't wrong, but it's incomplete. A 2017 analysis published in Agricultural History Review by Dr. Bruce Campbell of Queen's University Belfast found that maslin fields in medieval English manorial records consistently produced higher per-acre yields than monoculture wheat or rye fields on the same estates, even in favorable growing years when risk management wasn't relevant. The yield advantage persisted across good years and bad years. The farmers weren't just hedging. They were, without knowing the mechanism, exploiting the same mycorrhizal nitrogen transfer and competitive exclusion dynamics that the Lamberton researchers would document 800 years later. In West Africa, the intercropping of sorghum or millet with cowpeas, a practice that has been documented continuously for at least 2,000 years across the Sahel region, produces yield advantages of 30 to 50% over monocultures in the same environments. A comprehensive meta-analysis published in Field Crops Research in 2019, covering 226 separate studies of African cereal legume intercropping systems, found an average land equivalent ratio of 1.38, meaning that to produce the same total output as the intercropped system, you would need 38% more land planted to monocultures. Every time, across every culture, across every continent, the same biological logic kept reasserting itself. Diversity creates synergy. Synergy creates resilience. Resilience creates abundance. Now let's address the counterintuitive finding that surprised the research community most, because the Lamberton data didn't just show that 3 species beat 1 species, it showed that 3 species beat 8 species. And understanding why is critical if you want to apply this research practically. The 8 species mix in the trial, treatment 37, which included cereal rye, hairy vetch, crimson clover, oilseed radish, phacelia, buckwheat, sunflower, and cowpea, was the kind of diversity cocktail that has become fashionable in regenerative agriculture circles. Seed companies sell these mixes with names like Soil Builder Supreme and Diversity Blend Pro. They're marketed on the logic that more functional diversity means more biological services. That logic isn't entirely wrong, but it misses a crucial concept: functional redundancy versus functional complementarity. In treatment 37, crimson clover and hairy vetch were both fixing nitrogen. Cowpea was also fixing nitrogen. 3 nitrogen-fixing species in the same mix sounds like triple the nitrogen benefit. In practice, it created competition between the nitrogen fixers for the same mycorrhizal fungal partners, specifically for the rhizobium and bradyrhizobium strains that form the most efficient nitrogen-fixing nodules. When multiple legume species are competing for the same microbial symbionts, nodule formation efficiency drops in all of them. The buckwheat and phacelia, while beneficial in other contexts, were competing with the cereal rye for the physical space and light needed to create the canopy architecture that supports hairy vetch. And the sunflower, with its allelopathic root, exudates. Sunflower roots release chemicals that inhibit the growth of neighboring plants, a phenomenon documented in a 1996 study in Journal of Chemical Ecology, was suppressing the establishment of the very species it was supposed to be complementing. The 8-species mix wasn't creating 8 times the biological services. It was creating biological interference, species competing for the same resources, the same symbionts, the same physical space, and in some cases actively suppressing each other's most valuable functions. Treatment 23's power came not from the number of species, but from the functional complementarity of the 3 species chosen. Each species occupied a distinct ecological niche. Each enabled the others without competing with them. The system had no redundancy and no interference, just three species, each doing something the others couldn't do, each making the others better at what they did. This principle, that the quality of species interactions matters more than the quantity of species, is one of the most important and most frequently ignored lessons in polyculture design. There's one more variable in the treatment 23 story that the Lamberton researchers identified as critical, and it's one that even experienced cover crop farmers frequently get wrong. Termination timing. When you terminate a cover crop, whether by roller crimping, herbicide application, mowing, or tillage, determines how much of the biological wealth you actually capture versus how much you lose. The trial tested 3 termination timings for each treatment: early termination at 30% flowering, mid-termination at 50% flowering, and late termination at 75% or greater flowering. For most single-species treatments, the differences between termination timings were modest, typically 10 to 15% variation in subsequent cash crop performance. For treatment 23, the differences were enormous. Early termination, killing the cover crop at 30% vetch flowering, produced subsequent corn yields that were only 11% above the control. The mycorrhizal network had not yet reached its peak density. The nitrogen transfer from vetch to rye had been proceeding for only 6 to 8 weeks. The biological system was interrupted before it had fully expressed its potential. Mid-termination at 50% vetch flowering produced the highest subsequent cash crop yields, the 12.4 metric tons per hectare average we discussed earlier. This timing corresponded to peak AMF network density, peak nitrogen transfer rate, and maximum root biomass contribution to soil organic matter. Late termination at 75% or greater flowering produced lower subsequent cash crop yields than mid-termination, despite having higher above-ground biomass at the time of killing. The reason? At late termination, the vetch was beginning to redirect energy from root exudates and mycorrhizal network maintenance into seed production. The biological network was actually contracting as the plant shifted its investment strategy toward reproduction. Terminating at this point captured more above-ground biomass but less of the underground biological infrastructure. The practical implication for farmers is specific. In most Midwestern growing zones, this mid-termination window for treatment 23 falls between May 15th and June 1st when the mix is fall planted, or between October 1st and October 15th when spring planted as a summer cover crop. Getting the timing right is not complicated, but getting it wrong by even 2 weeks in either direction can cut the system's performance by 30 to 40%. This is the kind of precision that separates farmers who get results from farmers who get disappointed. Research plots are 20 meters by 20 meters. Real farms are hundreds or thousands of acres. Does the treatment 23 system scale? And if so, what breaks down at scale that works perfectly in a controlled trial? The honest answer is that scaling introduces 3 specific challenges that the Lamberton trial, by design, didn't fully address. The first challenge is seeding equipment calibration. The 20/60/20 weight ratio requires precise metering of 3 species with dramatically different seed sizes and flow characteristics. Hairy vetch seeds are roughly twice the size of cereal rye seeds. Oilseed radish seeds are smaller and rounder, with different flow dynamics through standard grain drill seed tubes. Getting all 3 species to flow at the correct rates simultaneously requires either a drill with 3 separate seed boxes and independent metering systems, or a pre-blended mix that has been carefully calibrated before loading. Several early adopters of the treatment 23 system in Minnesota and Iowa reported disappointing first-year results that, upon investigation, traced back to seeding ratio errors. One farmer was achieving a 40% radish, 50% rye, 10% vetch ratio because the radish seeds were flowing faster than expected through his standard drill. The resulting stand had excellent weed suppression in year 1 but failed to develop the nitrogen transfer network that drives long-term performance. The solution is a 30-minute calibration exercise before planting, running each species separately through the drill over a measured distance, weighing the output, and adjusting the metering gates until the actual delivery matches the target ratio. time-consuming once, worth it every year thereafter. The second scaling challenge is regional adaptation. The Lamberton trial was conducted in southwestern Minnesota, USDA Hardiness Zone 4B, with average annual precipitation of 650 millimeters and a frost-free period of approximately 150 days. In warmer, wetter regions, hairy vetch can become aggressive enough to suppress the cereal rye rather than being supported by it. In drier regions, oilseed radish establishment can be unreliable without adequate soil moisture. Researchers at Kansas State University ran a modified version of the Lamberton protocol in 2021 and 2022, substituting Austrian winter peas for hairy vetch in Zone 6 and warmer, where hairy vetch's aggressive late-season growth can create management problems. The Austrian peas maintained the nitrogen-fixing function while being more manageable in warmer climates. The composite performance advantage in the Kansas trial was 173% above the trial average, lower than Lamberton's 210%, but still extraordinary. The third challenge is economic, the seed cost for treatment. 23 is higher than for cereal rye alone. At 2023 seed prices, establishing treatment 23 at the recommended seeding rates costs approximately $47 to $62 per hectare, compared to $18 to $24 for cereal rye alone. That's a real cost difference, but against the $112 per hectare annual fertilizer saving and the compounding soil health benefits, the payback period is typically less than 2 growing seasons. The Lamberton trial results were presented at the 2023 American Society of Agronomy annual meeting in St. Louis, and the response from the research community was, by academic standards, unusually animated. Several researchers questioned the composite performance index methodology, specifically whether weighting all 11 measured outcomes equally was the right approach. If you weight cash crop yield more heavily than, say, earthworm density, the rankings shift somewhat. Treatment 23 still wins, but the margin narrows. The researchers' response was that the composite index was designed to reflect total agroecosystem function, not just productivity, and that the choice of equal weighting was a deliberate value statement about what soil health means. Others questioned the generalizability of the results beyond the specific soil type at Lamberton, a Hamerly clay loam with a native pH of 7.1. Would the same combination perform as well on a sandier, more acidic soil? On a heavy clay? The honest answer is probably not identically, but probably directionally similar. The biological mechanisms, nitrogen transfer through mycorrhizal networks, radish-enabled high-fall depth extension, AMF-mediated competitive exclusion of weeds, are not specific to clay, loam soils. They operate in any soil where the biological community can establish. But here's what the research community largely missed in their response to the Lamberton data, and what I think is the most important implication of the entire 6-year trial. The discussion at the ASA meeting focused almost entirely on the practical application question. How do farmers implement treatment 23? What are the seeding rates? What's the termination timing? What's the fertilizer reduction protocol? All legitimate and important questions. What almost nobody discussed was the theoretical implication that the 210% performance advantage of treatment 23 over the trial average cannot be explained by any single biological mechanism. It requires all 3 mechanisms: nitrogen transfer, mycorrhizal depth extension, and competitive exclusion, operating simultaneously and amplifying each other. Remove any one mechanism and the system's performance drops to roughly the level of a good single-species treatment. This means the system is irreducibly complex. You cannot extract the active ingredient and apply it as a product. You cannot replicate the outcome with a synthetic fertilizer and a herbicide. The only way to get the result is to create the conditions for the biological community to assemble itself. That is a profound challenge to the input substitution model that dominates even progressive agricultural thinking. It suggests that the goal of soil management isn't to supply what the soil needs. It's to create the conditions where the soil can supply what it needs itself. We've covered a lot of ground in this video. 6 years of data, 41 treatments, 11 measured outcomes, 3 biological mechanisms, thousands of years of human agricultural history that arrived at the same conclusion from the opposite direction. Let me bring it back to what matters. The Lamberton trial didn't just identify a winning cover crop mix. It demonstrated something that has implications far beyond which seeds you put in a drill. It demonstrated that biological complexity, when intelligently structured around functional complementarity rather than random diversity, creates outcomes that reductive single-variable thinking cannot predict, cannot replicate, and cannot replace. The 210% performance advantage of treatment 23 is not a number you can achieve by optimizing any single input. You can't get there by applying more nitrogen. You can't get there by using a better herbicide. You can't get there by selecting a higher-yielding cash crop variety. The only path to that number runs through the soil biology, through the mycorrhizal networks, through the nitrogen transfer conversations happening underground between root systems that we spent most of the 20th century poisoning with synthetic inputs and mechanical tillage. The indigenous farmers who planted Three Sisters combinations 2,000 years ago didn't know about arbuscular mycorrhizal fungi. They didn't know about nitrogen-15 isotope tracing or weed suppression indices, but they watched their fields for generations. They noticed what worked and what didn't, and they passed that knowledge forward. The biological reality they were responding to was the same biological reality that the Lamberton researchers measured with $200,000 worth of laboratory equipment. That's not a coincidence. That's convergent truth. Here's what I want you to take away from this. If you're farming, the question isn't whether you can afford to implement a system like Treatment 23. The question is whether you can afford not to. At $47 to $62 per hectare in seed costs, with a payback period of less than 2 growing seasons in fertilizer savings alone, and a soil health trajectory that builds compounding biological capital every year, this is not an expensive experiment. This is a long-term investment in the productivity of your land. If you're not farming, but you're gardening, the same principles apply at any scale. A bed-scale version of the Treatment 23 ratio, hairy vetch, cereal rye, and oilseed radish at 20/60/20 by weight can be planted in any garden bed as a seasonal cover crop and will build soil health faster than almost any other approach available to you. And if you're neither farmer nor gardener, but you're simply someone who cares about where food comes from and how the land that produces it is being treated, share this video with someone who needs to hear it. The farmers who are building soil health right now are making decisions that will determine the productivity of that land for the next 50 years. The more people who understand this science, the more farmers will have the knowledge and the economic justification to implement it. The soil doesn't need us to manage it. It needs us to stop mismanaging it. And sometimes, the most powerful thing we can do is get out of the way and let 3 species of plants do what they've been doing together for thousands of years. If you've made it this far, and I mean genuinely, if you've watched this entire breakdown, you're the kind of person who takes this seriously. That's exactly who this channel is built for. Subscribe and turn on notifications because every week we go this deep into the science that actually drives agricultural outcomes. Next week, we're breaking down a 2023 meta-analysis of 47 studies on mycorrhizal inoculant products, and the results are going to challenge some very popular and very expensive assumptions. Drop your USDA growing zone in the comments right now. Tell me where you're farming or gardening, and tell me whether you've tried any multi-species cover crop system before. I read every single comment, and your zone data genuinely shapes what we cover next. If you're in Zone 7 or warmer and wondering whether Treatment 23 translates to your climate, that comment might just become its own video. Wenli, Ren, Ra Ti Dobur, Randor Ra. We started this video with a number, 210%, and I promised you that by the end, you'd understand not just what that number means, but why it exists, what biological reality it's pointing at. The why is this: life supports life. Diversity creates resilience, and when you put the right species together in the right ratio at the right time, you don't get the sum of their individual contributions. You get something larger, something that builds on itself, something that the land has been trying to show us for thousands of years. Every time a farmer noticed that their mixed planting outperformed their monoculture and passed that observation forward to the next generation. The Lamberton researchers measured it. The Three Sisters farmers lived it. The question now is what you do with it. If this changed how you think about soil biology and cover cropping, leave a like. It tells us this depth of content is what you're here for, and it helps us reach the farmers and growers who need this information most. If the mycorrhizal nitrogen transfer network we covered today fascinated you, you need to see our breakdown of what happens to that network under different tillage systems. Click right here. The data will change how you think about every pass you make with a plow.